Anybody have any questions? No? Good. Lecture. I gave you opportunity. You did not seize that an opportunity. Therefore, you get the consequences of not acting quick enough. That's fair, right? I gave you a chance. You did not go quick enough. Therefore, the chance was gone. That's fair, right? That's fair, right?
No.
Not really.
Okay. Now, let's try it again. Does anybody have any questions about exam 1? No? Okay, lecture this.
What does ORF stand for?
ORF.
This is ORF.
ORF. You know what an ORF is? It's like the most important thing ever. It's the thing between the start codon and the stop codon. It represents the gene sequence. In an RNA, we call it an open reading frame, because it's a reading frame that's long enough that it potentially codes for a protein. It's a molecular term for when we sequence DNA. If there's a certain distance between a start codon and a stop codon and there's in the same group of 3, then that could usually be a protein. And the group of 3 idea is more important as we go forward, because we haven't really talked about translation and how it actually works. But the number 3 is important for keeping things in the right grouping. The open reading frame. Now sometimes it's a DNA issue, but a lot of times we talk about it as far as the RNA is concerned. The RNA contains an open reading frame or multiple open reading frames. We can't say the RNA contains a gene. That's no longer DNA anymore. So we kind of switch between DNA terminology to RNA terminology. But we do sometimes use ORF when we talk about DNA. Mainly when we haven't decided what a gene does, we don't know what a gene does, we call it ORF. Because we don't know that it actually does anything yet.
So that's why it's called open, just because we don't.
The open part means there's a number of codons between a start codon and a stop codon. And so when the computer looks at the DNA sequence, it says, here's ATG, here's, let's say, TGA, and they're the same reading frame, meaning the same group of 3, sort of part of each other. And there's enough codons in the middle, which is a protein. So you should hear much more about that in molecular biology if you take it. But for us, it just allows us to talk about organizational differences between prokaryotic organisms and eukaryotic organisms. Yeah, go ahead.
Can you go back over like the STR analysis and like how to identify what's the parent, and then like the graph or whatever?
Let's start at the very beginning. What information do we get from looking at just this idea? The first thing, STRs, short tandem repeats. It's a number of repeats in DNA that gives different alleles. We can measure or visualize STRs for individuals by doing what we call genomic electrophoresis. So we separate the DNA by size. And so number 1 doesn't mean that there's 2 of these pieces of what we call bands. When there's 2 of these bands, what does that mean? I mean, really heterozygous, right? You have one allele from one parent, one from the other. We don't know which ones yet, from which parent, but we have 2 different alleles. Then we have just one band. We call it homozygosity or homozygous, because again, there's only 1 piece of DNA there. But in 1 piece of DNA is 2 pieces of DNA, because you have to have 2 of everything. So that's the first thing you need to understand is sometimes you see individuals that are heterozygous, other times you see individuals that are homozygous. You have to remember that this one had to receive this same piece of DNA from 2 different parents. And so when we do the STR analysis, we're trying to decide between parent 1 versus parent 2, and there's usually some possibilities. And what we want to see is, does the parent have a band in common with the child? In this case, for number 1, there's 2 possibilities, the top one and the bottom one. You can't have both, because the parents are going to give half their DNA to their offspring. So this parent would either have this band here or this band here, indicating that it would be the parent of this child. Similarly, we're trying to see if that's also true in the other direction. trying to see if parent two has this or has this. Now you can't have a case where this would happen. Because that doesn't tell you who's the parent. Because both are not possibilities. Sometimes this happens where you see that individuals might have one overlapping piece of DNA. We have need another piece of information to usually say what's going on. So for parent one over here, they had another band down here. Parent two had a band up here. And now what is our analysis? Which parent gave us this piece of DNA? Parent one or parent two? Right. It has to be parent one because the only way to get this one would be from the parent two. And so by process of elimination, we see that this piece of DNA had to come from that parent, this piece of DNA had to come from this parent. And based off of what we see here, neither one nor two are the parents of child two because none of those have the DNA that we need for that particular child. And so you're simply looking at your child and trying to see where did this piece of DNA come from? Does it match a piece of DNA from a parent one, or does it match a piece of DNA from a parent two? And sometimes there's overlap. And so you can't just say number one, number two, because it doesn't account for the other piece of DNA that's present there. You'll probably see something like this on the exam on Wednesday, where you need to do a little thought. One might be easy to find, the other sort of a process of elimination after the fact. So this is the essence of the STR analysis, comparing DNA fragment sizes.
This is about like a different topic. Is
Okay.
that fine?
Well, let's see here, are there any other questions about STRs? Did you find? Okay. Then on to something different.
Okay, could you kind of like explain how the parents differ in the monohybrid cross and the dihybrid cross?
What does monohybrid mean versus what does dihybrid mean? That's the first part is mono means one, di means two. And so when we do a genetic cross, we're looking to see the inheritance of a trait in our offspring. So a monohybrid cross, only one trait is different. And so we look at the green pea plant versus the yellow pea plant and see the inheritance of green pea color versus yellow pea color, or purple flower versus white flower plant. And the dihybrid cross, we're looking at parents that differ in two traits. So one is purple flower with yellow seeds, the other is white flower with green seeds, for example. And Mendel was trying to show that the thing that controlled flower color was inherited separately than the thing that controls seed color, for example. And you'd only see that if you do a dihybrid cross to show that they are inherited independently of each other. So parents really don't change. It's just, they're all true breeding. It's just, if you're doing a monohybrid cross, there's one trait that's being examined. In the dihybrid cross, you're examining two traits. You even do a trihybrid cross, and now you're looking at the inheritance of three different things at once. You just get, each time you add another trait, you get an expansion of the 3-to-1 ratio. So maybe you can do 3x plus y cubed to get to the number of things we need to get there. But the way I like you to think, or the way I presented it during lecture, we tie together the monohybrid cross directly leading to the principle of segregation, whereas the dihybrid cross was more where the principle of independent assortment came from. Any other questions, anyway?
Can you go over the penetrance calculation? How to do it?
Oh, God. It's stupid easy.
It is, but somehow I
It is
still did it
easy to make a
wrong.
mistake. So, let's define penetrance first. Can you define penetrance, or would you like me to define it for the class? Or can anybody define penetrance?
One gene affects multiple variants.
What's that?
One gene affects multiple variants.
No, that's pleiotropy. A good guess. Thank you for finding. Go ahead.
Penetrance is the number of organisms whose phenotype matches their genotype. So whose genotype is expressed.
Yes. And so penetrance is directly opposite of the variable expressivity. And penetrance says, if you have this genotype, maybe you have the phenotype. And so we see, I don't want to call it unlinking, because we use linkage as another term, but again, the idea of unlinked. Sort of genotype and phenotype typically goes together. So the whole point of glycobiology is genotypes and phenotypes go together. But here, genotype doesn't always make the phenotype happen. And cancer disease is where we see this most often, where you can have an allele for breast cancer, but you don't get breast cancer. Or your likelihood of getting breast cancer is higher or lower based on what your DNA sequence actually says about it. And so we want to try to tie together to look at how often does the phenotype show up for a particular genotype. And there's typically only 3 genotypes to worry about: the homozygous dominant, the heterozygous, and the homozygous recessive. Now, for penetrance calculations, we sort of assume we can do whatever we want to. In practicality, some of these things won't actually work out. But we need to define the disorder. So, which would you like for the disorder to be in? This one, or that one? You one vote for that one. Anybody else want to vote for that one?
No.
Or what's somebody want to vote
Yeah,
for
no,
this
that
one or this one?
That one for sure.
Okay, we'll do this one. This one will have the disorder. So what do we call a recessive disorder? So anything that has this genotype has the potential to have the disease. And then now we need some numbers. Let's say that 50 of these individuals in the population, 50 of these individuals in the population, and 50 of these individuals in the population. Make math real easy for us. Maybe it'll make it different. Just for fun. 30. 20. Just, why not? And so first thing we need to know is how many people in the population have each of the different possible genotypes, which we have now. We know that there are 20 homozygous dominant, 30 heterozygous, and 50 homozygous recessive. But because we know the disorder is only going to affect individuals with the homozygous recessive, we don't care about these numbers at all, because they don't affect our calculation. We're trying to see how many individuals with a genotype have the disorder or phenotype associated with that genotype. But we still need to say one more number. We need to know how many of these 50 actually have the disease, and just to be easy, we'll do 10. 10 diseased individuals. Lepers on our colony. Question?
Wouldn't the heterozygous one count for it since it has the possibility for the recessive?
No, because you need this genotype to have the disorder. And again, we are ignoring the idea that the allele is there. We're looking at a particular genotype, which I know is going to be confusing for some people that have had other classes, but we are ignoring the other two because they don't have the exact genotype that we're looking for. But in practicality, there is a calculation that can be done with this number. We're not going to worry about it. We're just looking at how many people have this particular genotype. Because this is the genotype with that the disorder is involved in. In this simplistic model, now we just have to figure out how we do our calculation. And it's the number of individuals divided by the total number of individuals. So we get 10 divided by 50, which makes our penetrance 1 out of 5, which is 20%, or actually 20.0%. I like to be 3 decimals, 3 numbers. Technically, it's not 3 significant figures because zeros at the end don't count as significant figures.
It's after the decimal place.
But it's not decimal place. I guess
It's
it's
after
significant.
one. It's 30.
Here's only one.
Decimals?
Here's only one. I don't wanna get into chemistry, because it's not chemistry, we don't have exactly significant digits. But if there are not three numbers in your answer, then you're most likely gonna get a little snarky comment that says, follow the directions that say put 3 digits in your answer. Like some of you got in your quizzes. Because you went 4 digits, or 2 digits, or 8 digits. I'm like, come on. Make my life easier, make your life easier. Round. But that's what we need to do. We need to, again, the easy way to make a mistake is to use all of the individuals. Which is something we need to do in other calculations, but not on the penetrance calculation. Because again, we're trying to show how many individuals with a particular genotype are likely to have the phenotype associated with that genotype. So don't care about everybody, only a small subgroup of the total population. And so based off of this one group of individuals, we could extrapolate and say the penetrance of another population could be 20%. Maybe below the house. Are there any follow-up questions about penetrance or delineation?
Is the inactive X chromosome called bar body, or is it the single active X principle? There was.
Bar body is the name of the structure that is now present in the cell because of the crumpling of the X chromosome. So what we visually see after X chromosome activation is the bar body. Mary Leone didn't call it X chromosome inactivation. She called it the single X principle of what it was called. So that was her first idea on what it was called. Now we just call it by something a little more simple, more straightforward. X chromosome inactivation.
Can you explain how transposase works? Like, which does it cut out the transposon and then it cuts to insert the transposon? It does both of those things?
It all depends on if we're talking about a DNA transposase or a retrotransposase. But for a DNA transposase, we would have our piece of DNA, and there's a transposon, called the TN. So the first thing we need to happen is this would need to make the enzyme called transposase. And once the enzyme is made, it's going to cut this thing out. So you get a piece of DNA with a gap in the middle. This can be glued back together, or like this, but this can be pulled back together. And this can be put somewhere else. So now the transposase will cut sort of here and now put it into a new place.
And it cuts both strands?
Yes, we have to cut both strands because it's a double strand of DNA molecule that's being moved. Now for the retrotransposons, we skip the first step because we don't cut out the transposon. We make RNA intermediate that is made into a double strand of DNA, but eventually a transposase has to stick that thing into DNA. So at the end of the day, we're cutting both strands of DNA molecule to insert our transposon into a new location. It's just to sort of forget about cutting out. It's kind of a weird sort of case because it'll apply to both.
So then is this kind of the same thing, how like we said like transposons can like jump to different locations?
And the transposase that each transposon has is what allows it to cut itself out and to move it to a new location.
Okay.
Now physically it doesn't move it, like the transposon doesn't grab onto it and say, let's go over here. It just cuts the DNA, cuts it out, and then it cuts it somewhere else, and that's where it goes. And generally, if you think about the DNA inside both the nucleus and inside the prokaryotic organism, it's all on top of each other. So you don't have to go very far to cut out and move to a new location. It's usually they sort of go close to each other sometimes. Must be how much more time can we spend today? That's not on measure. Yes, figure.
Can you like try and explain to me like how karyotyping and like trisomy and then monosomy, all that connects together? Like how to distinguish? So I know karyotyping is used on both of those, but I don't.
I mean,
know
a
the-
karyotype just by itself is a representation of all the chromosomes that an individual contains. That's the first part. Basically, karyotype is a chromosome picture. So however many chromosomes you have, you see representation of all those chromosomes present. Trisomy and monosomy are cases where the diploid number is not present for every single chromosome that an individual contains. You either have one copy of the chromosome rather than two, or you have three copies of the chromosome rather than two. And because we're looking at all the pieces of DNA, you should be able to see, is there something by itself that doesn't have a partner? Or is there something that has two partners? So being able to see 3 versus 1, something that is easily done by looking at a picture of DNA. Now, to get into actually how do we make the karyotype, that's a significantly different question, which we don't talk about.
Yeah.
But once you do what you ever need to do to get the DNA out of the cell in the right context and the right format, someone or a computer puts the picture together. So we can see each homologous chromosome pair, see is there any abnormalities. Is one missing, is one extra, is one bigger, one smaller? These are the types of things we can see just by visual inspection. So think about, like, who's ever done those? Look at a picture, find the 5 differences. That's kind of the thing, is like looking here, what's different about this? There should be 2 of everything. If you see something that's not 2, that's a weird thing. Except if you're in the sex chromosomes, because then you just see either 2 X's or an X and a Y. But for the autosomes, the 1 through 22, there should be only 2 of each. A parent 1 copy and a parent 2 copy. So we're not gonna do anything with karyotypes and cuss them all. We're not gonna be asked to look at them or do anything with them. They know what it is and what it's useful for. See how much DNA an organism has, and can we spot aneuploid states like monosomy and trisomy, or can we spot chromosomal irregularities like duplication, deletion, or some other things? Like a copy number variation would be very easily viewed, hopefully, in a karyotype, because one chromosome would be significantly bigger than its partner. What else would you like to ask about?
Can we talk about the different types of epistasis?
No.
Okay.
There is really only one important thing that we talked about with epistasis. Well, first, there's 2 things. First is the definition. So the fact that multiple genes affect a single trait or phenotype is what we want to think about for epistasis. And epistasis and pleiotropy are opposite effects. So one versus affecting many versus many affecting one. What I was trying to illustrate with the different types of epistasis is show you that we do see some weird interactions. And so we typically are so focused on dominant or recessive, that dominant always beats recessive. But with the one case, the 12 to 3 to 1, it was recessive. No, that was right. 12 to 3 to 1 was a dominant affecting another dominant. You don't see very often where there's 2 dominants and they fight each other for dominance. But the 9 to 4 to 3, a recessive genotype was having some effect on a dominant genotype. So that would be the only thing you want to be able to remember is what's going on. In the 9 to 4 to 3, is it a dominant or recessive controlling something else? In the other case, the 12 to 3 to 1, what's going on? Dominant or recessive, what's controlling things? Now, if you comment overall, the other ones you'd see lots of different types of interactions where things can occur in weird types of ways. And it gets fairly confusing pretty quickly. In the 9 to 7 ratio, there's 2, if you're homozygous recessive for one of 2 things, you obscure the dominant completely. So it's not just one, it's 2 possibilities that can destroy a dominant. There are some weird things going on. Epistasis, 2 or more genes controlling a single trait, and sometimes it's a recessive genotype that just exerts control, sometimes it's a dominant genotype that exerts control. typically, whenever you have a dominant thing, you get more of the thing, which is why the 12 and 3 to 1 go to the dominant, and the 9 and 4 to 3 goes with recessive. What other questions we have?
So with copy number variations, is that when you have an entirely different amount of entire genes? Or is it just longer than STRs?
Well, first, STR are individual repeat sequences, so it's not a gene, it's just a small repeat piece of DNA. How big it is varies. But these repeats are located within genes sometimes, and you can make more of these repeats, and you get more STRs. A copy number variation is take a chunk of DNA, let's say 10% of the chromosome or 20% of the chromosome. It's now duplicated or deleted. That would lead to a copy number variation effect. And so are we affecting 1% of the chromosome versus 20% of the chromosome? It's kind of the idea that STR is 1% and CNV might be 20%, or might be some other variation with numbers. But CNVs are really big chromosomal effects. STRs are relatively small in comparison. And SNPs are single nucleotides. So single nucleotides repeats that could be 1,000 long, but CNVs are 100,000 to 1,000,000 long. Basic size differences there. That's why CNV falls into like a more monosomy, trisomy state. Because even though you don't have an entire full copy of the chromosome, you've got enough of it to screw things up. Now, there is a fun story with STRs, but it's not here, it's in the next set of slides. Or for exam 2 material. So things we have heard throughout the semester, so make you remember some of the stuff for longer periods of time. Yes?
Is there a difference between mutated paralogs and pseudogenes?
A pseudogene is a result of the first thing. So, evolutionarily speaking, if you have 2 copies of a gene, you only need 1. So one is potentially allowed to accumulate mutations, whereas the other one, there's a genetic pressure, a selective pressure to keep it working. Some mutations you don't want to be in there, so you can't really control what mutations have, but sometimes you can, I care about this DNA more than I care about this DNA. And so when you make a copy of a gene through duplication event, that extra copy can do its own thing. And through mutating the gene, you can either make the gene non-functional, turning it into a pseudogene, or that those mutations could change the gene's function of the protein to be a new activity or a slightly various activity.
And that'd be caused by, like, natural selection would cause it?
I don't want to get into that discussion right now, because that is not the purpose of this class at all, and I don't care about natural selection at all. Who cares? I care about bacteria. Bacteria don't like, or they grow so fast, who cares? There's always a selective pressure to keep things functioning normally, because that keeps the cells functioning normally. If you have this extra copy that's not needed to function normally, you can either basically blow it up so it doesn't work anymore, who cares, or you can allow it to change into something new. Even then, maybe it doesn't survive or it doesn't get passed on to the next generation, so maybe it's just a fluke kind of thing. Yeah. There's a lot of examples of this in nature when you do natural selection. Lots of different variations of things, but only one change gets passed on to the next generation because it's the most dominant thing or the most common thing and the most advantageous thing to see. So you might see, 9 arms and 8 arms and 6 arms and 7 arms and 4 arms. 4 arms, But only one of those is probably the one that's the most advantageous for that individual and whatever environment's in. And that's the one, hopefully those individuals are the ones that are successful in making and they pass whatever their DNA is on to their offspring. But paralogs do allow for evolutionary changes to occur, at least not the evolution changes new functions to possibly arise. Because you don't want to mutate your good copy, because you need that for something else. But the other copy you can allow to do something else. You ever see Star Wars: The Next Generation? Sorry, not Star Wars. Right track. The Next Generation. Any tracking? No. Well, it doesn't really matter. It's a science fiction trope. If you have a copy of yourself, you can do whatever you want to it, it doesn't hurt you. If you go on its own life, it might be different than you, or you could kill it. Or it could kill you. Who knows? But when you have an extra copy of something, it can, sometimes there is a selective advantage to keep it the same because it provides something useful, like the case we saw with that picture. The slide that was shown, we talked about copy variation, where sometimes having extra copies of a certain gene are helpful based on your diet. But for everybody else, they don't need those extra copies, so they're kind of lost or inactivated over time because they don't serve a useful purpose. Think about what was the first blood type? We didn't have 3 blood types to start with. We had 1. Probably A. A, B, who knows? Yes, who wants to be A, who wants to be B? Okay, we'll say A is the first one. And if we duplicate that gene and it mutates, we get the B allele. Or, who knows, maybe not. Maybe something else happens. So let's go. Topic. I don't want to talk about that. I don't want to talk about natural selection. That's somebody else's class. That's Dr. Fitz's class.
Like when we were talking about the chromosome structure, like for the P and Q, like can those be like interchangeable? Like could you have a P arm that's longer than the Q arm?
By definition, P is always short
It has to be shorter.
because P stands for petite.
Okay.
Q stands for large. No, you don't know the large? The Q is silent. You ever seen this French word? Never seen this French word? Oh, here's a new French word for you. Large. But I didn't mention this probably at the time, which is why there's this confusion, but the P stands for petite. So, no, not that, petite. But it's called the short arm because it's the petite arm. So, it's always the short arm. By definition, P is always the shortest part of the chromosome. So wherever that centromere is, you've got 3 possibilities for location, but doesn't matter left or right, really don't have any meaning. Whatever's the shortest piece is always the P piece. And here, I guess, maybe you have to guess which one's P, but if they're both equal, then they're both, one has to be P, one has to be Q. They can't both be P. That's where the statement, the phrase, mind your P's and Q's, came from this story. No.
Oh.
That'd be cool though. I believe the same case. And maybe somebody was minding their P's and Q's when they decided that we should call the arms of the chromosome P's and Q's. Maybe it was Hardy Weinberg that fucked everything up for everybody, P's and Q's. Why not peas and carrots? How much you buy lunch? What if you have this thing like this? A diacentric chromosome, which is the P arm in this one? Whatever one's the shortest. I know we typically don't worry about these because these are abnormal things. But for a normal chromosome, a metacentric, submetacentric, and acrocentric, there is definitely something we can call P as the shortest thing between telomere and centromere. It's a shorter distance than the other piece.
And acentric and dicentric are really bad for cell division, right?
Yes, because you need that centromere for proper separation of chromosomes during things like mitosis and meiosis, which we will talk about hopefully soon. Yes.
For variable expression, is the phenotype always observed? It's just varies, like, cause the genotype and phenotype are always together, right?
So, variable expressivity and penetrance are opposites. And so, yeah, with variable expressivity, genotype is, I'm just gonna use the word linked, it's not the best word, I can't think of another word for it right now. Genotype and phenotype always go together. Why it's called variable is the fact that the phenotype is variable. We always get the phenotype, but the phenotype is now possibly has different characteristics or appearances or types. For penetrance, the phenotype is not always present, but whatever the phenotype is, it's going to be the same phenotype if it ever shows up. But here, the phenotype is always present, it's just a question of how much of that phenotype do you have. And so, think about epistasis and pleiotropy as being opposites. Think about variable expressivity and penetrance also as being slightly opposites of each other. But we have to be careful because in both those, variable expressivity and penetrance fall under the same category of variable expression. And so it's the expression of the genotype that is either making the phenotype show up or how much of the phenotype shows up. If we get this difference, then it's not always a one-to-one correlation. So every possible way that we can break the golden rule of phenotype is directly related to genotype, probably out there. Genetics is fun and interesting and exotic. Yes.
What do the histone proteins do again? Like their overall function?
Histone proteins are involved in compacting the DNA so that it fits inside the nucleus.
So that was all of them? Or all of them have a part?
Well, as a general rule, that's what histone proteins do. They compact DNA. But we separate the histone proteins into two basic categories. The core histone proteins are H2A, H2B, H3, and H4. And those are the ones that form the ball that DNA is wrapped around. The H1 histone is the DNA binding protein that will go in the DNA between the balls.
Can it?
It doesn't connect them because the DNA is connecting. So we have basically what we call the core histone, or the core nucleosome, is this structure here. And the DNA gets, wrapped around this, and it goes over here and gets wrapped around this. The core nucleosome is where we find the 4, the 2, the 3, and the 4. Whereas the H1 is what's going to code this piece of the DNA. So the cores physically wrap the DNA in the ground itself. The H1 just goes the DNA between the different cores that are present.
And then, I didn't get this down, I'm not sure if we're going to go back. In bacteria, it says they don't have the histone proteins, they have what?
Bacteria don't have homologs to the eukaryotic histone proteins. They have other DNA binding proteins that serve a similar function. So we don't have true histone proteins in prokaryotic organisms. We do have DNA organizing proteins in prokaryotic organisms. And these serve the same function. Compact the DNA so it fits inside the cell. That's the basic function of chromatin in general, is a more compact form of DNA that will actually fit inside the structure of the cell or the nucleus. And there's lots of studies where they've taken the DNA out of the cell and measured how long it is. And it is more than 10 times longer than the actual length of a cell. And so the only way to fit it into the cell is to squish it really tightly into a ball. Hope that it stays together, because you need space for other things.
Could you explain histone replacement?
No. I don't think there's much explanation that needs to go on. It's a simple fact we want to try to remember. The H3 protein and H4 protein, at least H3 will be replaced in the core of the nucleosome at the centromere. Centromeres are going to be important for moving the DNA around during cell divisions like mitosis and meiosis. And the protein that we substitute is called SINPA. So we remove H3, add SINPA around the nucleus, not nucleus, centromere, around the centromere. The SINPA protein is the thing that's going to recruit the other proteins that are needed to move the DNA around. So without SINPA, we don't know where to attach these things to the chromosome anymore. So the other thing that's important is this only happens at the centromere, because the centromere is where we're going to do our attachment point. It's not a random point, it's a specific point in every chromosome where is that centromere. So some signal in the DNA says that here is where we replace H3, and nowhere else do we replace H3. But this replacement allows us to do the important steps of mitosis and meiosis. The other thing that we talked about with histone proteins is the role of H4 and the connection between cell division and the methylation state of the H4 protein. So for that case, as the telomeres get shorter over time, eventually they'll get short enough where it will signal cell division to stop. So as the telomere gets short enough, a signal is sent to methylate all of the H4 proteins on histones to say don't copy this chromosome anymore because we're trying to stop cell division.
Do we need to know what SINPA means, or just that it replaces H3?
We don't care what it means.
How does the haploid state ensure chromosome consistency from generation to generation?
I think you said halfway.
No, haploid.
No, I got it after a second. Halfway. That doesn't matter for exam one.
Okay, well, it was on that question, the list of questions.
From 10A or from 10B? Because
It
we
was
talked
for the
about-
third quiz, I don't know. I thought it was.
I don't think so.
The
Is
third
that-
quiz, like list of review things. You know, the review questions after.
It's on
the
10A.
slides.
Is it?
Mhm.
Where?
I
I don't believe you.
Sorry.
I don't believe you at all. Oh, you were fun with me. Seriously, that's in 10A? I don't think that's right. Where's it say? Let's see. Let's see, right. I don't see it in 10A.
It might have been 10A, but it's somewhere on the sheets for exam one. Okay.
This sheet here says exam 2. 10B.
Well, we don't have to go over it if we don't have to know it for the test.
So on this question, where is it? Let me see it. I don't see it on this listing here. And it's not in those two files, so I don't know where it is. Regardless, or as I like to say, disregardless. Extra here regardless. What's the question again? Never mind, I got it. When it comes to sexual reproduction, which is a exam 2 topic, not an exam 1 topic, the whole idea of meiosis is to take a diploid cell to create haploid cells because we need the fertilization step to happen next. And for fertilization to work correctly, to maintain the chromosome number from parent to offspring, be half our DNA, then we add another half to it to get back to a whole. So for humans, as an example, we take our 46 chromosomes and split them up into 23s, give 23 to offspring, get 23 from another parent, rebuild the 46. So by going from diploid to haploid and then back to diploid, we maintain chromosome number from parent to offspring and through the subsequent generations. That should be exam 3, exam 2 thing, not an exam 1 thing. Who's ready for a lecture, 4 minutes of lecture? I know you are looking forward to lecture, aren't you? Look at all these things we get to talk about. I'm going to update the slides, that's what I just still said last semester. Ready? 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 Set? What comes next? What? No.
Go.
Go is what comes next. You have trouble picking up by putting out.
Go back.